Everyone to 2 days of the Scanpy course. I'm Maren Büttner. I'm postdoc at Fabian Theis's lab, and I have been working with single-cell RNA sequencing data analysis for the past 5 years during my PhD and also during first year of my postdoc. Okay, first of all, I give you a brief overview on today's workshop. So I first give you an introduction on the basic workflow. Then we go into the first Scanpy session in the breakout rooms where we set up the environment. Whoever had still problems with the environment, we solve it there. We load the data, we install additional packages, and then we're ready to go. After the first session, we make a short break, and then we go into the second session, starting with quality control and data pre-processing. I should give a brief introduction to this as well. And then we're already at the lunch break for 1 hour, from 12 to 1. Then we look at normalization and batch effect correction, which is a really important topic when it comes to single-cell data. And we should spend quite some time on. Then again, after that really, let's say, complex topic, we have again a break, and then finally we go into the fourth session where we have our data ready and go into annotating the data, and potentially we also look into pseudo time, and then the day is almost over already, and then I would like you to take some time for a bit of a feedback and also a summary and also a bit of a discussion until 5. Okay, so once upon a time we had a bulk genomics. Well, we still have bulk genomics, but when you think of bulk data, it's always like making a smoothie. So you have many different cell types in your body, and they have different compositions. And this is like taking different fruits, and you would and then you mesh them up. So you mesh up your cells, you mesh up your fruits, and all you get afterwards is some kind of smoothie of cells or here of fruits. And once you take a sip of a smoothie, you realize you have the dominant taste of an apple or a banana, but you miss to taste the kiwi or the berries that you added to your smoothie. And it's the same with RNA sequencing in the bulk state. So when you have a bulk profile, you only see like the dominant cell types, and you tend to miss rare cell types. With single-cell data, you have access to all the cell types and all the characteristics of each cell type. So you can basically sort your fruit salad and see how many cells you have and what's their characteristics and also what's their composition. We use this pipeline called Droplet sequencing, and this is depicted here in this video, where you shoot the single cells into nanoliter droplets, and each of these droplets is a small reaction volume. So and this technology helps us to upscale the whole process of single-cell RNA sequencing to thousands of cells. And at the same time, it reduces the costs tremendously. So here's a small diagram of how many cells have been sequenced and published in several experiments over the past years. Well, we stop here at 2017. And you see that a huge upscaling here from 2014 already until 2017, and the scale just didn't stop there. Nowadays we have Atlas projects with millions of cells. That's the new normal today. Usually when you start to do your own single-cell experiments, with the 10x platform you go with, let's say, 3,000, 5,000 cells at a time. Okay, let me give you a bit of an overview on the experimental aspects of single-cell RNA sequencing. So you start with sample of interest, you generally have to extract the RNA from these cells. So you have these RNA molecules floating around in your reaction chamber, and then you have to convert them into cDNAs, and these prepared cDNAs are then sent to a sequencing core facility usually. These cDNAs are sequenced, and then you end up with millions of sequences that you then finally have to map to the genome and do your data analysis on. So this is the experimental part, and we will only focus in the end on the data analysis part, but I would also like to cover a bit on the genome mapping, at least theoretically. I already mentioned the 10x platform. We have several ways to do sequencing, or not sequencing, but library preparation of single-cell RNA sequencing protocols. So here I depicted like 6 different protocols that are like mostly available and mainly used. They differ, of course, and they also have different purposes. So with the 10x platform, so that's a droplet-based platform that you have, so the second column and the orange one. You have the microfluidic system. You can sequence thousands of cells, but you cannot really measure any additional factors on this. So if you want to do a FACS sorting, and you want to know which of the cells has a certain FACS index, you would probably want to go with the MARSeq protocol, the ScriptSeq, or also probably this the SmartSeq protocol, because there you can sort the cells by index, and you keep additional information on the cells. However, if you sort cells by index, you usually use plates to sort them and to extract the RNA, and then you get less cells. That's one part. So how many cells can you get? What kind of information can you get from the cells? There's another thing that you have to take into account once you want to design such an experiment. There's, on the one hand, we have full-length protocols where we capture the whole transcript, and we have the 3' bias protocols where we only capture the 3' end of the transcript, which is closest to the poly A tail. So on the protocols I depicted here, you see that some of them are full length and some of them are not. So SmartSeq is usually a full length protocol, while the others are 3' bias protocols. The second thing, or the third thing that you have to take into account is if you want to use unique molecular identifiers, short UMIs. Those you can add to every molecule that you capture, and that helps you to reduce the noise in your data. Why would you like to reduce noise in the data? I show you on the next slide. So when we capture our cells, let's focus here on the Dropseed pipeline. We isolate the cells in our reaction chamber. Then we do reverse transcription to create cDNAs, and here we add the UMIs to our cDNA molecules. So that's a short barcode. Usually it has 8 to 10 base pairs and identifies every molecule that we can capture. Then we synthesize a second strand, and finally, to increase our signal, we have to amplify our molecules by cDNA amplification. Usually we use here a PCR, so polymerase chain reaction, to create multiple copies of our cDNAs, or there's also the so-called IVT protocol. So, and once we have gone through the amplification step, we can construct a library. So, and here is the amplification step is critical when you talk about noise in single-cell RNA sequencing data. So when you have the UMI, you can tell apart a copy, so a real copy of a molecule that has been present in the cell from a technical copy that has just been created during the amplification step. So because in the amplification step you create hundreds and hundreds of copies of the same molecule, and only with the UMI you can in the end tell if it's if you have 10 copies of the same molecule, if it was present in the cell in the original during the reverse transcription, or if it has been created during the PCR step. In the SmartSeq protocols, you can't tell apart the technical from the biological copies, and that's why you have much more noise in the data. So just to, once you think of different protocols to work on, that's something you have to reminded of. Okay, so we have constructed a library. Now we map the transcriptome to the genome, and well, after sequencing, and what the sequencing core facility usually does, it creates so-called BCL files, and they're then transcribed into FASTQ files. Sometimes BCL files and FASTQ files are considered both as raw files. However, when you want to submit data or raw data to a public resource, they request you to take the FASTQ files or to submit the FASTQ files. So when I want to talk about raw data, it's usually the FASTQ files. FASTQ files are huge, so you shouldn't store them on your laptop. Rather use a storage unit. That's the part of advice that I should give you here. And you should always keep them, because these are the files that you would like to go back to once you want to remap your data. Here in this picture you see the Cell Ranger denoted. So Cell Ranger is a software from 10x Genomics that comes with the 10x Genomics library prep system. So they also have the software that helps you to map your FASTQ files, so let's say the sequences of your data, to make a count metrics. At the center we have a bioinformatics core facility that does the mapping for you if you ask them to. All right, what is this mapping process? So when you have your transcripts or your cDNA molecules, you would like to know to which gene they actually assigned to. So which part of the genome is actually represented by your transcript. And this is the so-called mapping process. So you have the short pieces of DNA, and then you have to map it to the genome. Again, this process, I would say it's still some kind of part of high performance computing, so I haven't tried it on my laptop. I always went to a server when I wanted to map data. All right, just briefly, this is how a FASTQ file looks like. So you always have like the first line that starts with the .at. Then it's followed by a sequence identifier and an optional description of the file. Line 2 is the raw sequence letters that has been read out in the NGS core facility. Line 3 starts with the plus and is some, again, some description of the data. So they're somewhat important to assign the cells, assign a read to a cell, but you don't have to look into these kind of FASTQ files one by one, because you create thousands of them, and let's say these identifiers are designed to talk to machines rather than people. But I want you to have a look at it at least once, and that's that. Okay, let's go back to data. So now we can go into the details of single-cell RNA sequencing. A bit of would like to show you one property of single-cell RNA sequencing, which is the so-called sensitivity. Don't be shied away by this huge plot. You see there's at least 20 different protocols around that do some kind of RNA sequencing, and I think so. This plot is already from 2017, and I think there's already a couple more protocols being published ever since. Okay, so what you see here is a so-called molecular detection limit. So how many copies do I need to detect this gene or this transcript at least once? The leftmost violin plot shows you bulk samples. So in a bulk experiment, you need at least 10,000 copies, something between 10,000 and 100,000 copies of the same molecule to detect this gene at least once. All the other protocols are single-cell protocols, and you see there's a huge difference across these protocols, but they're all more sensitive than the bulk protocols. So some of these protocols even go down to detect one molecule. So molecular detection limit is one molecule, meaning every molecule that's present is captured. But that's a rare case. These super sensitive methods, like usually this is a SmartSeq protocol that's super sensitive, they only have few cells. So we had that, we discussed that problem already before. If you, so either you have high throughput protocols or you have low throughput protocols. SmartSeq is one that is super sensitive in contrast to Dropseed, Chromium, 10x genomics. These protocols have high throughput, but they're more shallow. So when you go to the right-hand side of the plot, you probably see the Chromium violin plot is like the fourth on the right-hand side. This is the 10x genomics protocol. So we see that the molecular detection limit is about 100 molecules. So we talk about, so this is more of a shallow protocol, but it has a high throughput advantage. So overall, you can't have it all. So high sensitivity plus high throughput plus full length plus low noise level doesn't exist. So choose two, and then you already have the protocol that you should use. Most of the time, the shallow sequence protocols give you all the information you can possibly detect from your data, and they tell you everything you ever wanted to know. However, if you go down to study transcription factors, which are low in copy number in a cell, then you would rather go to a SmartSeq protocol, which is more sensitive. But then we already talk about a targeted study of cells, which is a perfectly fine way to go, because it depends on the question that you would like to answer with your data. The Atlas projects, at least I am mostly involved with, go with the Chromium device, so the 10x platform, because you have the high throughput and you can capture rare cell types rather easily. Okay, so now let's leave the experimental session, and I would like to introduce you to the basic workflow. So we made the paint from library prep to sequencing to mapping, and now we have our count metrics. And now we can analyze the data. So a count metrics, that is the output of, for example, the cell ranger pipeline, is a matrix that has the dimension cells by genes. So we have about 30,000 genes, and we have about 3,000 cells that come from a single 10x experiment. So this is the count matrix. And every entry in this count matrix is the number of RNA molecules we have detected. Again, dimension 30,000 by 3,000 is huge, and you don't want to check every line one by one, but you would like to discover patterns in the data set. So in order to discover meaningful patterns, you have to go through the process of pre-processing, filtering your data, normalizing, and let me show you a couple of steps that you have to do here. So first of all, you do a QC, so quality control, where you distinguish whether your cells are actual cells or if this is debris. You normalize the cells for cell-specific bias and cell type-specific bias, also for gene length if you have a full-length protocol. Then you select features like highly variable genes. So that means the variability of the gene is higher than expected by the intrinsic noise of your experiment. Once you have done that, you go for batch effect correction. So that is a correction for the systemic bias across samples. Pre-processing takes usually 80% of your time, and all the rest of the workflow is about 20% of the time. So don't be scared if this pre-processing takes longer than you think. Okay, once we've done the pre-processing, we end up with a gray map. So this is a two-dimensional representation of our data. So we went from 30,000 dimensions to 2. So we boiled down all the information that is in our count matrix to 2D. Okay, and now we have to make sense out of this data, and we do this in the downstream analysis step first by a clustering step. It should show up like right now. Yep. Okay, so clustering means we group the data. So we group our cells by their similar properties, and then we can annotate these clusters and this is usually done through a process using marker genes. So we use prior knowledge to see if a certain gene is expressed, and this is what we use to assign a certain name to a cluster. Okay, these steps are almost always performed in a single cell setting, and then you can ask more questions in the downstream analysis, but that depends on the kind of data set you have. A popular question is so-called trajectory inference. So if you have, for example, a stem cell, so like as in this case, you have stem cells and you have mature cell types, you would like to know how the stem cells change to become the respective cell types. Where do they undergo fate decisions, for example, and which genes are involved in driving a certain decision to go either to cell type A or cell type B. So using trajectory inference and pseudotime, you can uncover these kind of processes, and you can, when you sort your cells along a certain trajectory, you can even uncover the genes that drive a fate decision from one cell type to another. Furthermore, when you have different conditions in your data, you would like to know what is changing in a certain cell type upon disease versus healthy. So this is a typical question that you ask for in differential expression. So which genes change across groups? What you can also ask with single cell is, how do compositions of my cells change in different conditions? So, for example, if you have a certain perturbation, for example, a disease model, usually have an invasion of immune cells in a tissue, which are not present in a healthy sequence, and you can nicely capture the changes of these different cell types' appearances using single cell data, and this has not been accessible before in this large scale. So so that's just a small outline on what you can do with single cell data. There's always more. There's always more details. If you would like to read up on this whole pipeline workflow a bit more, I strongly recommend to read this paper from my colleague Malte. That's called Current Best Practices in Single Cell RNA Sequencing Analysis. It also comes with a tutorial that you can try out on your computer, and this course that we are doing today is also basing on his course. Okay. Let's dive into Scanpy. Scanpy works with the so-called NData package. It's a so-called annotated data frame. It has the following structure. So first of all, we have a data matrix that's called X and has dimensions cell by genes. And alongside with the data matrix come annotations. And we can make annotations on the observations, so for every cell. For example, how many genes does this cell express? How many counts did I find? What's the cell type? And so on and so forth. So that's some metadata that you add in this ops object. But you can also annotate the genes. So what's the mean gene expression? For example, do I have an alternative gene name? Is this gene highly variable? That's all added to the variable annotation. And furthermore, you also have this unstructured annotation. So in the unstructured object part of the data frame goes everything that doesn't fit into the other two categories. So, for example, the result of a gene expression test, colors for a plot. These things go into the unstructured. I'll show you an example how an NData object looks like. So this is one that you probably will create yourself and during the course today. So what you see is you have an NData object, because I can use my mouse here. So it has the dimension NOP, so 1000 cells, and it has 12,000 genes. Then the metadata are listed. So in my observations, in the ops, I have counts, I have the number of genes, I have the mitochondrial fraction, and so on and so forth. More factors that all apply to the observations. I have annotations for the variants. I have in the unstructured, I have which cell has which neighbors. I have colors for Louvain graph. I have results of a differential expression test. What I haven't listed in the picture is the ops-m object, also the var-m object, and layers. So the ops-m object contains our low-dimensional representations. So we have a PCA object, we have a t-SNE object, a UMAP object and diffusion map, and a force atlas, so a graph object. In the VRM object we have PCs, so principal components, and finally the layer. The layer here is called counts, so that means it's a it has the same dimensions as our X object, so it's n-ops times n-vars, but it has different entries. So here we have the counts, meaning 1, 2, 3, 4, 5, so the count of every molecule. But the X matrix has potentially a different normalization, or it's batch corrected. And probably sometimes you don't want to look at the batch corrected data, but you would like to go back to your counts. So the unnormalized version of your data. And this is what you can save in the layer. Okay, so this structure will accompany you during the course. So that's why I stress this so much. But by finishing with the stressing, I would like to go to the hands-on session. So from raw count metrics to cell type annotation. And this is a couple of things that you should think of when you use your Docker container. So sometimes you have to deal with memory issues, and especially the more complex tutorial that I created works well with only 60 gigabytes of memory and 1 gigabyte of swap. The simple tutorial takes less memory, so don't freak out when your computer doesn't have that specifications. Okay, when you use Docker container, increase the swap to the maximum. In the settings, you can do that. Free the memory as often as possible. You can do that by del and then the object name, and then it's flushed off your memory.